Hello, my name is Isabela Espino-Marquez. I am a third year junior here at the University of Oregon studying neuroscience with minors in Latinx studies and Spanish. And today I'm going to be talking a little bit about my summer research that I have done within the Action Control Lab under mentorship of Hayami Nishio and Ian Greenhouse. It's been an amazing experience working under their mentorship. I started in winter term of last year, and prior to my summer research, I worked to get a better understanding of the dynamics of our lab and more so our mission. And throughout the term, I came to the understanding of our lab's primary um, aim is just to better understand how the brain controls movement, specifically the dynamics between one's motor performance and its relationship to neurophysiological properties in the motor system. We hypothesize that to successfully complete a task, one is dependent on excitability states of the motor system located in the motor cortex and other associated regions of the brain that control movement. And we intend to test participants' ability to suppress unwanted movement, cancel ongoing movement, and broadly suppress the noise in the motor system to facilitate movement. And so this summer I had the amazing opportunity to continue my work in the Action Control Lab. Primarily, this looked like getting more familiar with our coding system, analyzing our protocols, and presenting relevant research within our lab. And so one of the first tasks that I was given was familiarizing myself with MATLAB, which is the coding system we use within the lab to record participants' data during data collection. And I began familiarizing myself with the server and took onboarding courses to better understand like simple logistics, as my experience using code was super minimal to none. And this was really interesting for me to navigate its specific properties and coding services, and it was really helpful to understand how things were oriented and more being able to troubleshoot smaller errors. But prior to getting into any protocol, I worked on a lot of smaller tasks, just helping myself to better understand the basics of what our lab participates in. That first step was getting like a service to how to download MATLAB on our computers and creating this sort of sandbox and participating in that sandbox, which is just where novice learners can kind of learn how to trim and cut data sets in order to better understand what we're looking for in the data and how to better analyze data sets. Luckily, luckily I did have an amazing GE who was able to kind of craft this, and I was able to create another document that had more of these like boundaries and rules and a matrix for students to log data sets and make sure they're properly analyzing it. And this wasn't a large part of my research, but I did find that it was crucial to better understand our lab's aims, just to get a better understanding of why and how we do things in the lab. And it's a task that's usually given to undergrads, so it feels a little bit tedious, but also it's super helpful to kind of getting that data set understanding to see the actual participant involving. But once following participation data collection, I learned more about the requirements and technicalities of conducting quality research. So for like example, making sure that we stay within a 4-week time frame to ensure consistent results with our participant, and conducting around the same type of day, and making sure participants are right hand dominant, and making sure they're still eligible for MRI and TMS, and checking for family history or for implants, and doing this to ensure that our data sets that we're getting from participants is a good example of the general population, since that is what we study, and making sure that we get reliable, consistent data is really important. And so once participants have been scanned, we are able to schedule with LCNI for imaging, and follow all MRI protocol. A large part of what I get to do is making sure that we adhere to like all procedures, and that looks like participants are comfortable and um, feel okay with being a part of it, and making sure they're not wearing any ferret clothing, and just ensuring that they understand what they're going to be going through, just since the MRI can be pretty loud and it's a small space. But during the MRI scanning, we do many different types of scans. So we do localization, T1 anatomical, diffusion, right M1 and left M1, OCC, and T2 anatomical, all of which collect anatomical and physiological data to ensure proper identification of certain areas. Anatomically, this offers us a greater visual of the motor area in the frontal cortex, like the primary motor cortex and occipital cortexes on both sides of the brain. And these are the primary areas we study within the lab lab to better understand areas that are associated with suppressing unwanted movement, canceling ongoing movement, and broadly suppressing noise within the motor system to facilitate movement. And physiologically, we just ensure that nothing abnormal is occurring within these regions for participants. But yeah, so once those MRI scans are done, we upload each image into BrainSight and Polaris, which are the primary data tracking devices that we use within the lab, and this allows us to precisely align and calibrate each participant's brain image within the software, recreating their positioning accurately and streamlining the start of the session, and having everything preloaded ensures like the smooth transition when participants arrive and allowing us to maximize the time spent on actual testing and data collection. But beyond these preliminary tasks, my main focus of my research was really diving into our newest protocol, which represents the foundation of what I'll continue to work on throughout my undergraduate studies, and I have gotten to get a really great understanding of what we do. So with the following of this video, I'm mostly just going to be talking about our protocol. So before each session begins, we just make sure that participants are consented and reaffirm that they're eligible and comfortable with participating in the study. Once we have that, we're able to load MATLAB, and which is what we do to record data, and we collect electrometography, and then to get those readings, we connect electrodes to specific muscles for stimulation using transcranial magnetic stimulation, which is TMS, and this allows us to hotspot first, which is we're testing for the right FDI, making sure that we can contract that muscle, and that controls the right pointer finger. The process of hotspotting helps us locate the motor box, which is the exact area where stimulation elicits a response in the FDI muscle. Once the optimal spot is identified, we're able to perform thresholding to determine the minimum stimulus needed to activate these nerve clusters in the FDI, which ensures precision in stimulating only the relevant neurons for our study, which is considered thresholding. But following that thresholding, we register the participant with BrainSight, aligning the TMS coil positioning with MRI calibrated data, and this step allows us to maintain consistent simulation parameters across the rest and trial tasks, where we mark anatomical landmarks, including the nasion, and two specific MRI base points where we place vitamin capsules during, just to have that marker, aligning and making sure that we are accurately testing throughout our studies. But once that setup is completed, we stay within MATLAB's configured boundaries and begin stimulating different positions around the hotspot, creating a detailed hand knob map, which is the area in the motor cortex responsible for hand movement. And by mapping these various regions and orientations, we capture the topography of the hand's motor representation. This mapping is similar to creating a geographical map, as each part of the body has a corresponding area in the brain with the hotspot that we find, representing the primary control center for the right index finger. However, due to overlapping in the motor homunculus, this region isn't perfectly distinct, especially in athletes who often have more complex overlapping within muscle control areas. So we stimulate areas surrounding this hotspot, which helps us assess each participant's unique hand representation and investigate potential correlations between representation size, GABA levels, and performance metrics such as reaction time and EMG activity. We also vary stimulation orientations as neural populations respond differently to specific current direction. This comprehensive approach gives us a fuller understanding of cortical excitability in the hand region, allowing us to identify neural populations involved in various functions, from intentional movement to relaxed states, and even responded to unexpected stopping, which is something that our lab is really interested. And so once we've collected that data for both when participants are at rest or when they're participating in the task, we initiate data analysis by cleaning and organizing all of the information from these data sets. And this involves trimming and isolating all of the MEPs and burst responses, as well as discarding any trials that can't be analyzed due to noise or inconsistencies. And so each trial for the rest task is roughly 150 trials and 500 task trials for substantial foundation for analysis. Following this preparation, which is a lot of what I get to do, is just trimming that data and making sure everything is concise. Graduate researchers are able to perform statistical analysis to kind of redefine those boundaries. But we're continuing to test participants under these conditions to ensure consistency and reliability in our data, and I'm very excited to continue testing new participants. I'm extremely excited and very grateful to continue working on. This will very likely be what I continue doing for the rest of my undergraduate career. So incredibly grateful for this amazing opportunity. But just to highlight a couple other things that I worked on in the summer was getting to work with other undergraduate students and presenting to our lab. For a short time, I was working on a SICI protocol, which is short interval cortical inhibition, and sharing that at a lab meeting was a really great highlight of how that worked out. But to conclude, I found myself continuously inspired by the continuous and ever-growing process of research and understanding that it's completely dynamic, and making sure that I truly understand what our lab continues to study is very exciting and an amazing opportunity that I'm so grateful to have. Moving towards the future, my goals include potentially writing a thesis regarding what we study in our lab. But yeah, I would just like to say thank you so much for this amazing opportunity. I could not be more grateful. Thank you so much.